introductions, so then we're all here for the first session. So we have Keynote, Mr. Robert Kennedy from the University of Michigan. So we all know, so we'll start. So Robert Kennedy is the Hobart H. Willard Distinguished University Professor of Chemistry, Professor of Pharmacology at the University of Michigan. He earned a PhD at the University of North Carolina. Wait a minute. Bob's been here before. You all know Bob, right? Who doesn't know Bob? No. So people don't know Bob. One thing I learned this week, Bob has a Wikipedia page. You know you're someone when someone on Wikipedia makes you a page. All right, so I joined Bob's lab at the University of Michigan back in 2003, but I joined his lab in 2004. Bright-eyed, bushy-tailed, and ready to do science. I knew that I shared a common passion with my advisor, a desire to invent analytical chemistry methods to study biological applications. Just like Bob, it was not until graduate school when I realized that I could legally marry my passion for neuroscience and analytical chemistry. One interesting quirk I learned about Bob early on was that he did not like to tell us where he was going. He would just disappear. Maybe he thought us 20 graduate students and 6 postdocs would run amok and get nothing done. However, we were always on to him. Johnny and Jim would open up Internet Explorer and ask Dr. Google where Bob was. It didn't take too long to find him. Bob always asked us for slides for the zillions of talks he was invited to give. I think I had one of the fewer number of inquiries for slides. In the 5.5 years that I was there, I think Bob asked me once or twice for slides. However, I was in the audience at PitCon when he flashed one of my slides, and I cheered in my head for my 30 seconds of fame that day. Bob has a unique presentation style. His slide decks are like a photo negative of the default PowerPoint template. Black background, white text, blue italic titles, like what I see right here. Brings back memories. At the time, I never really understood why he did this. But by the end of my tenure, I had embraced his style because I saw it put less stress on the eyes to read. It wasn't until I joined the ranks of biopharma when I had to conform back to the boring white background, black text. Though I did use a Bob template for my first metamine-funded poster here at CE Pharm. I will end with one memory that really stuck in my mind from my graduate experience. It was a day where Bob and I both learned a life lesson. During my second year in graduate school, Bob invited one of our arch rivals to present a seminar to the department. The Kennedy lab was quite competitive with several labs in the field. Curiously similar to how biopharma is currently competing to get the next GLP-1 version 2.0 medicine to market. Adrian Michael was notorious for publishing work that questioned the scientific veracity of using microdialysis as a sampling tool. Microdialysis was the foundation of all my projects in Bob's lab. I took this to heart, and at the end of Adrian's talk, blurted out some sort of question comment that turned a lot of heads. I made it back to the lab, went into Bob's office, and promptly broke down crying. Bob's face was as blank as a bright, new spiral notebook. That day, I taught Bob the power of emotions. He gained his footing and talked me back from whatever cliff I was hanging off of. He told me it was honorable to feel so passionate about my work. He emphasized that you need to defend yourself and your work. However, be sure to check your words and put on a dampen switch every once in a while. So that day, I formally earned the name Pitbull because I was passionate about my work, I would latch in, and sometimes I don't let go. That was also the day that I broke Bob. Going forward, he was less regimented, more easygoing, and he told us when he'd be leaving the office to go to conferences. It is with great honor and privilege that I introduce Professor Robert Kennedy to give the keynote today. Thank you.
No, it really is an honor to be here with the CE Pharm crowd and Cass have great memories of working with CAS over the years. Even ran a conference that CAS did many years ago when they had microscale bioseparations. And by the way, I ran that meeting in Vancouver, and it was not on fire. It was raining cats and dogs. So, maybe we work well together. No, it's a real honor to be here. And the biggest honor really is having Mohammed and Kristen both up here, since they're both former people in my lab. And it's great there's nothing better about being a professor than having students in your lab and watching what they do, and then seeing how they grow and develop and the kind of careers they develop later on. So that's just one of the best things about the job. So today I want to tell you about some work that unfortunately will have very little actual analysis of pharmaceutical entities in it, but hopefully it'll give you some thoughts and ideas about using CE in different ways. And this has to do with using affinity interactions in capillary electrophoresis. And what I hope to do is sort of showcase the variety of ways that these two things can work together, that is affinity and CE. So I want to set the stage here just by pointing out, that affinity interactions has a very important place in separations, and you can think about it as a way to add selectivity to an assay. We think about things like Western blots and Southern blots and affinity LC. You can also think about it as important for purifying compounds, as in affinity LC. It's a way of measuring binding constants for different interacting partners, and it's also a way of discovering interacting partners, like think about the electrophoretic mobility shift assay. So affinity interaction plays a big role in separations. I mean, there's a Nobel Prize represented in some of the things I mentioned there, as well as numerous discoveries made with these kind of technologies. Now, CE itself is unique among separation methods in some of its properties, and so as you imagine, there's some interesting new ways you can use affinity interactions in separations. And maybe the most unique one is this concept of something called affinity CE that George Whitesides first described. So if you're familiar with this technique, it means we separate a compound, we get its migration time, and then we add a potential binding partner to the migration buffer, and we observe a shift in the migration that's associated with the amount of binding that happens. And then you can add a higher concentration, do that again, and ultimately develop these shift curves, and from that you can elucidate binding constants. And I kind of don't like the term because it's a little too general, but it is a very cool kind of technique. So then affinity probe CE, which is what I'll talk about most today, is where we actually let the equilibrium happen in the mixture and then separate the bound and the free partners and detect those. And I'll show you different ways that we can use that. There's something called non-equilibrium capillary electrophoresis of equilibrium mixtures, or NESEM, that Sergei Krylov developed. And here what we do is we allow the binding to happen, then when we do the separation, we allow some dissociation to happen during the separation. And this gives a modified curve that you can analyze to pull out kinetic and binding constants. And of course, there's a variety of Western blot formats now available in the CE type format as well. And I'll talk a little bit about that later today. So there's lots of ways that you can use affinity with CE separations. As I said, I want to spend most of the time talking about this use of affinity probe CE. And what we mean by this is we're going to have some target analyte. Sorry, the mouse is only showing up on the screen down there, so I got to, there we go. Have some kind of target analyte, and then we're going to add an affinity probe. This is some kind of binding partner. Normally we'll have a fluorophore on it. We allow them to bind, and then we separate them by electrophoresis, and hopefully fast enough that there's not any dissociation that happens. And what we should be able to see by fluorescence detection is the free affinity probe, and then the affinity probe bound to that complex. And so we can use this in different ways, as I'll show you. But first of all, just there's been a huge variety of binding interactions that have been looked at by this, which are listed here. And I'll try to highlight a few of these as we go along today. But it really shows the versatility, I think, of using this affinity probe idea. So some of the earliest things that were done with affinity probe assay, and I should point out that Barry Carter was the one that coined that term affinity probe. We were calling it immunoassay because we were only using immunoassays at the time. But that's, affinity probe is a good general term, I think, for this. So here's just a few examples of using this just for assaying a particular target in a complex mixture. So here's using an aptamer for IgE. You can see sort of the classic thing that we get in the electrophorograms. You have the aptamer there labeled with the fluorophore. Once we've now added the sample to this, so this is done in a plasma sample, you can see the IgE new peak here, which is the complex, and then even more Ig spiked in, and we get a bigger peak there. So you can sort of see that. You can use that as a quantitative tool to look at IgE, and you get the power of the separation here, and also the speed of this. If you compare this to something like an immunoassay, it's quite fast. Here's some example from Barry Carger's lab where they actually figured out how to label a Fab fragment with a fluorophore so they could use that to bind to their target. So you have the affinity probe here and then the complex in this peak here. And this is for detecting human growth hormone. And another trick that they use is they use isoelectric focusing here. I've been using CE. And what that gets you is that ability to concentrate the sample as well. So they got really good detection limits, down to picomolar detection limits for this. And in the lower trace here, the one nice thing about IEF is it was easy to separate the different forms of human growth hormone bound to the antibody fragment. So there's different versions of human growth hormone here, variants, that they're able to pick out and detect in this particular trace here. So these are just some examples of just simply using this as an assay tool, okay? So one of the things that we got interested in is expanding this idea and trying to look at protein-protein interactions. So just to sort of discuss this a little bit, most proteins interact with other proteins in order to actually perform their functions. And so you can actually build up networks of interactomes like this, where you see each of these nodes would be a different protein, all the potential binding partners, and all the different binding affects how these cells function in the cell and whether they do their function. And it's kind of an interesting idea to think if you could go in and selectively inhibit some of those interactions to be able to control cell function, be able to track it, for example, in chemical biology studies, or to inhibit it maybe for a therapeutic benefit. Now, one of the issues was, if you think about proteins interacting like this, is they were often thought to be undruggable because you have very, very large interaction interfaces, and a small molecule going in there and disrupting that big interface seemed like something that would be hard to do. But actually, people have figured out how to do this in some cases, and so there's a number of small molecule modulators being developed now for protein-protein interactions for various drugs, as various drugs. So there's kind of a nice emerging area here for pharmaceutical development. So if you want to develop such protein-protein interaction modulators and drugs, you need assays that can sort of detect this in screens. And so here's a few things that have been used before, listed down here, all very, very good techniques. If you look up here, I sort of listed out all the things that would be ideal in your method. So you want something to be really fast, low sample consumption, ideally label-free, easily move from protein to, sample to sample, different systems, and maybe look at multi-protein complexes as well. And while all these techniques are really good, I think it's fair to say that none of them sort of hit all those criteria there. So I think there's still room for development of protein-protein interaction assays. So if we come back to this affinity probe CE idea, it seemed like it would be a good one for exploring for protein-protein interactions, and because it's got a lot of the properties which I have listed there. So the first thing we did in this space was look at an SH2 domain. So you may know that SH2 domains are domains on proteins that will bind when a target protein or binder protein gets phosphorylated. And this helps direct cell signaling in cells. Once that phosphorylation event happens, you get another binding event, and that starts some kind of signaling cascade typically. Now, one of the nice things about the SH2 domain binding is that it kind of has some sequence specificity. So it will look for a particular peptide within the protein and maybe 4 to 5 amino acids and then that phosphorylation, and that's what it really binds to. So you can kind of cheat a little bit and look just at the peptide binding to the SH2 domain. So that makes life a lot simpler. And so here's an example of doing this where we took a peptide which we're able to label very easily and then allow it to bind to this particular SH2 domain from the Phin protein. And you can see here that as we add more of the Phin, we're able to get that complex peak being detected and the free peak is diminished with that. So easily detected, and that's in a very quick 10-second type separation. It's very little dissociation happens here. If you look really close, you might be able to see that this baseline doesn't go all the way back down to baseline, so there may be some dissociation happening during that. But from this type of data, you can generate binding curves. Another thing that you can do is discover or characterize inhibitors. Here's an example of adding an inhibitor to, so we have our complex here and the free peptide, and then we add more and more inhibitor, and you see less and less complex, more and more free there. That says unknown. We eventually figured out that was due to mild fluorescence in the inhibitor that we were adding. But again, you can use this maybe for a screen to discover new inhibitors, looking for changes in the bound-to-free complex, as well as characterizing inhibitors that you have. So as I said, that was a little bit of cheating because the peptides are very easy to work with. They're easy to label, they electrophoresis well, and so on. So we also started looking at more challenging kind of situations. Wanted to look at whole protein-protein interactions. And this is when we got maybe a little more than we bargained for by talking to Jason Geswicki, who was a professor at the University of Michigan and now is at UCSF. But he was studying the chaperone protein systems. So a little bit of background on this in case you're not familiar. So things like proteins like HSP70, which stands for heat shock protein, and HSP90 over here, these are proteins that will bind to unfolded regions of client proteins, they're called, and they help either refold them or send them off to proteases where they would get degraded. And their function is often modulated, that is the HSP functions are often modulated by binding to partners like HOP here or BAG3 there will modulate how these chaperone proteins work. And these chaperone proteins are very important for maintaining proteostasis, so maintaining the appropriate complement of proteins in the cells, helping get rid of misfolded or unfolded proteins, or helping refold proteins in cells. And you can have disruptions in that in things like cancer, where you're producing a lot of protein for cells, and also protein folding diseases also have disruptions in this process. So there's a lot of interest in thinking of ways to modulate this process, both from a chemical biology point of view, and also now there's actually drugs that block HSP proteins for treating cancer because it can help slow down the protein production. So we wanted to see if we could use capillary electrophoresis to study those type of interactions and maybe help screen for new interactors and so on. So here's an example where we managed to label HSP70 and get a reasonable peak. I say manage, one of the difficulties is always getting few enough labels in a reproducible enough way onto a protein and purified that you end up with a nice narrow peak in the electrophoregram. And so this is an example here with HSP70. And then we add the binding partner here and completely wiped out that peak, and there's a new peak over here. And just in case you're not convinced that that's actually the complex peak, we can go back and add back in unlabeled HSP70. That'll compete to bind to the BAG3, and it'll give you some free HSP70 back. So you can see now the free peak and the complex together in one electrophoregram. And again, that's a nice, quick 12-second separation there. So you can do quantitative things with this. For example, if we titrate in more HSP70, we get the changes in complex and free as you expect, calculate a % inhibition, and from that get binding curves and binding constants for this type of interaction. So that's a nice thing you can do pretty readily with CE. One of the things that we were curious about is if we could actually, would this be stable enough and reliable enough to use for drug screening type things, where you're trying to discover new inhibitors of that interaction? So we actually went through the whole process. We got a small library from our drug discovery center, and then did about 9,000 electrophoregrams, because we did these in triplicate on those drugs and looked at the bound-to-free ratio, and then put all that in a database and sort of figured out what hits we had from that library. And before I show you any results from that, I want to point out that we weren't doing this in a vacuum. We wanted to compare this to a more standard and widely used technique. So Jason's lab was using something called flow cytometry protein interaction assay. So how this works is you take a biotinylated version of HSP70, put it on a streptavidin bead. You let that interact with the bag 3, which now has a fluorescent label on it. Put that through a flow cytometer and detect the fluorescence on the bead. If there's an inhibitor present, it would knock some of the bag 3 off and you'd have less fluorescence, okay? So then they get binding curves from that as well. So we screened the same compounds by both assays, and what we found is that they both found some hits. CE found fewer hits than the bead assay, or the flow cytometry assay, but of the hits we found, if you test them different ways, like how often they confirmed by subsequent dose response curves or could be reconfirmed on the other assay, we found much higher reconfirmation rates with the CE. So what that tells us is that the CE assay is less prone to false positives than this other fluorescent-based screen. So you can ask, why is that? Well, I think there's a couple of reasons for that. One is if you look at sort of the setup for both of these assays, in this case, both the proteins are modified. That's going to create some possible issues with binders. There's a surface involved here that also is a potential issue, whereas here we just have the complex and only one of the proteins is labeled. So I think you have a more natural system here and less likely for things to go wrong. But I think the bigger issue is that with CE, you get more information. You don't just have a fluorescent readout, you actually have an electrophoregram. So you can look at that electrophoregram and make sure it looks like it should. So we know that the electrophoregram should have a complex and a free peak, and just the ratio should be changing. But if we see something like this, we know something went wrong when we added the potential drug to it. A lot of these libraries have things that are aggregating agents in them, and they can cause all kinds of funky things happen to your proteins. If you just get a fluorescent readout, you don't know that, but with the CE, you can see it. Some of the compounds are fluorescent themselves. This one's pretty strongly fluorescent, and so that also would interfere with the fluorescence-based assay, but not with the CE assay, or at least you could tell it. So these are reasons why this is a more selective assay. So I think it's got some potential there. It's probably not quite fast enough to be a primary screen for screening extremely large libraries, but certainly good enough, I think, for being a secondary screen to help rule out false positives with other assays. So everything I've been describing for you kind of makes it look like, oh, it's very easy. We take the binder, we get it labeled, we add the binding partner to it, and we separate them, no problem. And it's actually almost never like that. It's actually quite hard. There's a few things that can happen when you're trying to develop these CE assays. One is that the complexes can dissociate during the separation, so you need to go fast enough that you outrun that dissociation. That's usually not too much of a problem. Other things that can happen, you can have a small change in mobility. So the ligand binds to the protein, and it just may not move the mobility enough that you get a good complex separation. And then maybe the biggest problem is sometimes these proteins don't play very well, and they absorb to your fused silica or whatever. And of course, there are lots of ways to deal with protein absorption in CE, but many of them are not compatible with maintaining a protein-protein interaction. For example, you might say, well, I'll use high pH. That's my favorite trick. It works almost every time. But that's going to interfere with a lot of protein-protein interaction assay. Denaturing conditions, same thing. So you kind of have a bit of a conundrum there where it can be hard to develop a new assay. And this just shows you our first attempt with the HSP70 bag 3, that student adds the bag 3 and we get that sort of lump and we're like, what's going on? So it takes a while to figure that out. So we then started playing around with a different concept for looking at these interactions, and this is cross-linking the proteins before we do the separation. So we call this cross-linking capillary electrophoresis, or PIXE. And the idea here is that we allow the binders to happen as normal, but then we treat that with something like formaldehyde or glutaraldehyde. And now we should freeze in that interaction as it was in solution. We can now denature and separate by size. So that gets you a few things. You can, because the complex is covalently attached now, you can run whatever conditions you want, whatever it would take to get the separation. If you use SDS page type separation on the capillaries, then the ability that, most of these proteins are a good size, and when you bind them together, you get an even bigger complex. So now you can separate based on that size. You don't worry about small changes in mobility so much. So that's the idea, but does it work? Well, first thing we found is it was almost dead easy to develop new assays with this method. Here's a whole bunch of complexes that are, protein-protein binders that Claire was able to develop assays for, and none of these took more than a couple of days to work out the conditions. So this is a KIX with MIB25, so this is actually a transcription factor and a binding partner. Here's antibody-antigen binding with a difficult protein lysozyme, and then here's some of these chaperone proteins with binding to each other or to different different partner proteins. So you see all these were able to be developed pretty readily, get decent looking electropherograms. And then the next question is, are they quantitative? And again, we can get these same sort of complex binding curves as you titrate in a binding partner. Looks great, get KDs. And then we're able to compare these for all the ones that we tested to other non-cross-linking methods, and we found that we got a very strong correlation between the two, really good agreement in most cases. So it appears to be something that can work for a pretty wide range of binding constants. This is 10 micromolar down to 1 nanomolar here for binding constants. Then we also looked to see if these we could inhibit those with, with small molecule inhibitors. So these are some example of adding a drug inhibitor. So this JG, I told you my collaborator is named Jason Guestwicki. So this is what he called his drugs, JG with different numbers. And so you can add increasing concentration, see the increase in binding by this PIXI method. We compared it to the flow cytometry assay, got good agreement between the two assays for these different tested drugs, different hits. And then another thing that we did, and this is, I'm not a biochemist, but I started to feel like a real biochemist when I was doing this because Jason was giving us different modified bag with different modifications in it to try to map out what amino acids were important in the binding interaction. So as you change those amino acids and then you do the binding partner, the HSP70 is being changed. So you had the wild type binding curve with bag 3 and then these modified versions, and you can see that weakens the binding here. And so you're able to pick that out by this PIXI method as well. So lots of different things can be done with this. One more thing. Everything I showed you is once we did those separations and we were doing them on a regular capillary electrophoresis system, we were doing this sort of a minutes type separation. Those of you that know me know one of the things I like to do is try to go faster with separations. I hate waiting around. And also faster means you can maybe use it for screening. So we took that same concept and translated it to a chip. And so you can see that we can get that down to a couple of seconds for the separation of the complexes by size. And so then the idea was, could we use that for screening? And the answer is no, because the way we do this on a chip is you have a chip, you put a little sample in a reservoir, you get a quick separation, and then you got to empty out the reservoir, put a new sample in. Okay, so that's kind of a slow process. You're not going to do any kind of high throughput work with that. But Claire and I figured out a way to do this. So we had been working with droplet microfluidics. So in this type of system, what we do is you can take samples, aqueous samples, as maybe nanoliter volume, separate them, compartmentalize them with an immiscible fluid like a fluorinated liquid, which is represented by the yellow here. Excuse me, the yellow in this diagram. I'm not sure why he keeps wanting to do that. And then we just replace we take a tube and pump it into this, slide it into our chip, and I'll pump those droplets right next to the separation channel, the inlet of the separation channel. And the idea is that this oil that we use, this fluorinated liquid, is lighter than the aqueous, and so it just floats up to the top. And the aqueous sample comes out, and for a brief moment, it's sitting there right in the front of the electrophoresis channel with the voltage on, it gets pulled in, okay? And so we don't change anything here when we're running this. The voltage stays on all the time. The droplets keep going in, and we're hoping to get one injection after the other due to the droplets going in and next to that channel and then the oil pushing it out of the way. I can show you a video of this. It is, you can't see the whole process, but here you see the droplets right here. And then if you look over here, you'll be able to see the oil sort of floating to the top of the little reservoir here. So the droplets are just pumping in, and they're going into the inlet, and then the little oil comes out and floats to the top of that reservoir. Okay, so each time that happens, there's a new droplet, new sample coming in, being separated. And over on the right, you can see that this is not quite as good as sort of what we were doing before, but here's a 2-second free and complex sample being separated. Here's a droplet that had an inhibitor present, or sorry, this is free to bound, and then here's with an inhibitor present, there's more free. And here's just a whole string of these going out for over about 20 minutes or so. Several hundred injections, several hundred different inhibitors being tested in that brief period. So it can be a high throughput method and is extremely simple to set up. And like I said, I showed you what we were interested in, what we were working on. I'll just point out that other people have sort of demonstrated that protein cross-linking can be used for things maybe more relevant in what a lot of you would do. But this example, looking at antibody aggregation using that cross-linking idea and then quick separations on chips. So what I hope to have showed you so far with this is that this PIXI method and Affinity Probe CE can be used to determine binding constants, map interactions, rank small molecule inhibitors, discover new modulators, and can even separate multimeric complexes. Actually, I didn't show you that, but here, this trace shows you that. This shows you how nasty these things can be. So this is HSP90 binding to CYP40. So you just mix those two things together. You get a lot of complexes, okay? You have free HSP90. You have 1-to-1 of this. The HSP90 can bind to itself, so you can get what we call 2-to-0. And then CYP40, you can buy 1 or 2 of those. So you can have all those different complexes formed from that one mixture, and you can resolve them all by electrophoresis, which is kind of cool. Okay, so that's sort of what I would call direct analysis or affinity probe assay. We also have looked at using competitive immunoassays, and this is actually the first thing that we did. And I sort of already talked about it when I talked about the way when we look at these inhibitors for binding, but just to make it a little more clear if I went too fast before. So we're gonna now, and I'm gonna, since everything I'm gonna show you is about insulin, so I'll just call it insulin. So we have a mixture, it's got insulin in it, represented by the triangles. We can add fluorescently labeled insulin to that. We can add an antibody to insulin. Both of those will compete to, both forms of insulin will compete to bind. And if we separate by CE with fluorescence detection, we should see the free labeled insulin and the labeled insulin bound to the antibody. But the ratio depends on how much unlabeled insulin was present in the sample. So we can quantify that way. Now, the real reason we wanted to do this was not to analyze insulin in blood or something like that, although people have developed lots of competitive immunoassays to look at drugs, especially drugs of abuse and illicit doping by CE in different kind of urine and blood samples. But we were interested in the very high speed of this and how we might be able to use it for us as a sensing platform, okay? And so I want to show you this chip that sort of the culmination of this that we were able to develop. So we were interested in being able to monitor insulin being released from the cells that it normally releases from. So these are beta cells. They're found in the pancreas, and those pancreas are found in little clusters called islets. So the idea is we could put our cells in a reservoir on the chip. We can pull some of that fluid, we can perfuse that with different media, pull that off by electrophoresis, electroosmotic flow. We can mix in the reagents we need, allow them to react. Unfortunately, these reactions happen very quickly on the chip. And then do a separation down here, and we're going to do one separation after the other. And this way we can monitor the insulin being produced or released in this chamber. So here's just a picture of this single islet. This contains about 3,000 cells sitting in the bottom of that reservoir. And there's a little channel there where we're going to be pulling fluid off for analysis. And here's just showing you that we can do a sequence of electropharograms very quickly. These are every 10 seconds by this chip-based method. And these are just showing 2 different concentrations of insulin going in. So that shows you that the reactions happen fast on the chip, and we can quickly do this. And the reason we wanted to quickly do this is to monitor the dynamics of insulin secretion. They turn out to be quite interesting. If you take a single islet, put it in the chamber, and then we up the glucose level, so that stimulates insulin secretion. And I'll point out looks like a continuous trace. This is actually the insulin calculated from each immunoassay and then just plotting that as a line. Okay, so there's thousands of electrophorograms represented in this data. But you can see you get an initial burst of insulin release. And the very weird thing about this particular system is that even with the glucose staying constant, it doesn't stay elevated or go back down. It sort of oscillates on you, okay? And so this is a property of islets that they have this oscillatory secretion. This is the strongest one that we got, admittedly, but it shows you that what can happen, and you can monitor those dynamics. And then when we lower the glucose back down, it goes back down, returns to baseline. So you can monitor these dynamics of insulin secretion from individual islets. And we did many studies on islets and collaborated with many people to look at how we could do this. Here's one example of a collaboration that we did with Orman McDougald at the University of Michigan. He was interested in this LXR beta receptor. So this is an oxysterol, it's called liver X receptor, it's an oxysterol receptor. It's been well characterized in liver, but it's also present in beta cells and fat cells and other cells. So they knocked it out, and they found, surprisingly, that there was that the mice had what appeared to be a diabetic-like phenotype. So they wanted to know if there was a problem with the beta cells. And so we put those islets into the chip and compared them to wild type. And sure enough, when we added glucose, this is one concentration, then a higher concentration, you can see much less secretion on average from those individual islets from these guys compared to the wild type. So this example that you can use this for very routine kind of biological studies and discover things about these cells. So I kind of want to tie this also back to another trend that many of you are probably aware of, this idea of putting bodies on chip or organs on chip. So that work we were doing, we didn't call it organ on chip because that term hadn't been invented yet, but that's basically what it was. It's one islet on a chip, right? But some very cool work's been done where you can combine multiple tissues, for example, liver, lung, oxygen, and I think they've done fat and other things here on a chip, and then use the chip as a little circulatory system. And the main real use of this has been in the area of pharmacokinetic type studies, where you could put a drug in and watch its metabolism happen, mimicking what's happening in the body, right? So this has been one of the things that's been done, although there's other applications of this. Now, one of the things that we realized in looking at this very cool literature is that if they wanted to analyze what was going on, they still collected fractions out the end of the chip, and then analyzed them by something like LC or GC or something like that. And we knew that we'd already integrated assays into a chip, so we thought it might be kind of cool to do this. So we didn't do something as fancy as this, but we were interested in a type of interaction in relation to diabetes, which was a fat cell interaction with beta cells, okay? So, many of you are probably aware that obesity is a risk factor for type 2 diabetes. So the higher % body fat you have, the better chance you have of getting type diabetes, and it's just an amazing correlation. So one possible reason for this is, there's probably many reasons for this, but one possible reason is that adipocytes secrete a lot of stuff. And that gets into the bloodstream, and maybe some of that is not great for the beta cells. So here's a list of some things that they release. Fatty acids, so that's one of their jobs, is to provide energy in the form of fat when you need it. But they also are basically now recognized to be an endocrine type tissue and release lots of different hormones and other things, okay? So the question is, what does all this do to the beta cell? So we made a little chip here that is just a modification of the chip I showed you before. Everything to the right of this is what I've showed you already, but now we added some chambers for putting in fat cells. And then we were able to perfuse the fat cells and their secretions over the beta cell like you might have in a circulatory system. So here's just an example, some data from this. The bottom trace here shows you the average of several islets being treated with elevated glucose. So again, this should cause insulin secretion. You get that first phase release, and then it kind of calms back down. If you treat these cells with fatty acids, this is known to enhance secretion. So we added 50 micromolar palmitic acid, and you get this nice increase in insulin secretion with that. If you had 100 micromolar palmitic acid, again, this is one of the secretions from a fat cell, you get an even bigger burst, okay? If we put these in with some adipocytes, we get this really huge enhancement in insulin secretion. And we were measuring the fatty acid release from this as well, and it was between 50 and 100 micromolar of fatty acids coming off. So I showed you what the fatty acids did down here. The adipocytes are releasing somewhere down here, but their effect on the insulin secretion was way greater than that. And this shows that either the mix of fatty acids that they release or some of the other things that they're releasing are really affecting the beta cells and driving insulin secretion to very high levels. Now, the idea is that this is very acute, right? So under acute conditions, this is what happens. But over time, that very hard driving of beta cells is believed to lead to beta cell dysfunction and less insulin secretion, which would be bad and lead to diabetes. So this is something I've really wanted to do, is to continue this work and look at longer-term release, or longer-term interactions, but we haven't been able to do that yet. But it does show you can sort of discover things about this interaction that weren't known before. So the competitive immunoassays I've shown you, they can be extremely high throughput. I had one student, Kendra, who also operated chips for up to 24 hours, so you can do really a huge number of assays on these things. Integrate it with microfluidics, look at complex samples, and get all kinds of binding constants and so on. So again, a pretty versatile tool. I want to briefly talk about now Western blotting, because I won't talk about this too much because most of you are extremely familiar with this. You know, it's such a ubiquitous method, and also now that there's commercial products, it's a little bit different situation. But I just want to remind you that it's a traditional Western blot workflow is pretty slow. A lot of manual interventions, typically the way it's practiced in a biochemistry lab. But we now have some commercial products that can do basically the same thing. So Protein Simple is the most famous one here, where what we do now is do essentially a capillary separation and then immobilize the protein not by transferring it to a membrane, but by photo cross-linking it to the wall of the capillary tube. And then, where did my little arrow go? Yeah. And then you can, once the proteins are immobilized, you can pump antibody through there and secondary antibody to get your detection within the tube. So, you know, it takes advantage of a lot of properties of CE. It's automated, it's miniaturized, it's fast. So a very cool thing, really changing the way people do Western blots. Amy Hurd's group has done a lot at Berkeley, has done a lot of interesting things as well, I want to point out. Here's an example where they made a chip that had 48 different electrophoresis channels on it. Or, excuse me, 96 electrophoresis, no, 48 channels on it. Put sample in each one, ran them all simultaneously. They did also now cross-linking to the gel and then exposed to antibodies, and photo cross-linking to the gel, and not instead of the wall in this case, to get, and then pumping antibody through to get the Western blot. You can see the Western blot here for beta-gal and ovalbumin in this particular example. So we were also working in this area, and our idea was a little different. Instead of using photo cross-linking, we were going to take a chip or capillary, we've done both, and perform the separation, and then drag that chip along the membrane, and then let the transfer happen as the proteins escape from the elute from the chip, and then just let them bind to the antibody. So we get rid of that transfer step by letting it happen during the separation, and we take advantage of the faster separation you can get in chip or CE format to do this. And you know, what I'll show you, I hope, is that, you know, these other things are commercialized and are really great, but maybe there's some interesting things we can do with this as well that are unique to it. I won't go into all those details, but so this just shows that you can actually transfer a ladder. These are fluorescent proteins. We separate them on the chip and then transfer them to the membrane. And then you can look at the membrane this way if you want. That looks like normal sort of Western blotting or membrane type capture stuff, or you can do a line scan and get this sort of trace here for the size-based separation. So you can see that that looks fine. Actually, we did a study on this to see how much band broadening we got due to that transfer step. And So we detected proteins within the channel, which is the narrowest line there, and then within the sheath flow, which is the transverse part, and then once it's on the membrane, see how wide they were. And you can see that, there's a little bit of broadening that happens once you get on the membrane, but it's not bad. And what this tells you is that once those proteins come out of the chip and they hit that membrane, they bind really fast. They don't diffuse around on you and give you a lot of band broadening. So that was important for us because that means that this can work. So here's an actual Western blot with this approach where we're looking at ERK 1 and 2, just showing you can get decent resolution. These two things differ by 2 kilodaltons. You get a nice separation of those peaks. There's a line scan through it. And this is actually 2 injections done, one after the other, so you can see both of those. So that is just showing you that we can do this Western blotting this way. Detection limits can be pretty good. Here's an example of looking at actin, beta-actin. This is a peak for 9 picomolar here, so probably right at the detection limit, I think. I think my students said the detection was 8 picomolar, so, just above it. But point is being quite low, and given the amounts that we were injecting, that's just a few femtograms of protein. So one of the things that I thought was kind of cool about the way that we were doing this, because we weren't cross-linking to the gel, is potential to look at multiple proteins from one sample, okay? So in some cases, people have done what they call stripping analysis with Western blot. So they maybe separate a sample and get a gel or blot for it, as you see here. And then they would treat it with essentially surfactants to remove the protein, and then the antibody, and then come back and treat with a second antibody and do a second immunoassay to look for a different protein from the same sample. So you would do this if you were worried about not having enough sample to keep running. Now, it's not a very practical thing to do. People don't do a lot of this because you do lose protein going from with the stripping, and it takes a long time to do all the immunoassay again. But what we recognize with the CE method is, one of the disadvantages of the chip CE is you put a few microliters into these reservoirs on the chips, but you then only inject a few nanoliters. So what do you do with the rest of that? Well, what if you wanted to do multiple proteins, okay? So we could lay out a lane of one injection, move the chip over on the membrane, lay out another lane, and another lane, another lane, and then treat all those with different antibodies, for example, to look for different proteins. And so here's an example of doing this where we detected 11 different proteins twice from one 800 nanoliter sample on this chip. So you can see all these different, the ladder on the left, and then multiple injections for different proteins labeled here. Some of them were phosphorylated and unphosphorylated forms of the same protein, and so on. So I think there's potential for the multiplexing with this. That's kind of cool. How far could you go? Well, we haven't, what I showed you is the most we've actually done, but we did just ask if we just did repeated injections from one of these chips, how many injections could we do before things started to break down? So you might know that the buffer can start to break down from the being applied if you don't rinse out the reservoir, or you might deplete the sample over time. And we were able to get up to about 40 injections without much change, and then up to 60, you started to see a little shift suggesting the buffer was changing on you. So potentially you could go very high multiplexing. And then the ability to do that sort of gets at the throughput of this for the multiplexing. If you just do one assay and you do 2-minute separation, and let's say we do some kind of fast immunoassay, it's something that we've worked on and is also commercially available, it's like 32 minutes per. But if you're doing 62-minute injections, that's 2 hours, and then the immunoassay can be done at the same amount of time, it's down to like 2.5 minutes per assay. So the ability, when you do more of these injections, you take advantage of that, you can go have a higher throughput. There's a few other things that you can do from one chip. For example, you can have multiple sample inlets going to the same injector. And so then you could inject one sample, lay it on the membrane, to come back, run another sample, lay it on the membrane. Here's just an example. I'm not sure why anybody would ever look at egg whites and cell lysate in the same sample, but it's a demonstration of looking at 2 different samples and multiple injections to getting replicates for it. And then we did want to try to play the parallel channel game. This was something very hard, and my student refused to do it after doing it a couple of times. But you can see that you can cut multiple of these little points into the chip down here, and then drag all that along the surface and do multiple samples at one time. So a couple different ways you can use these chips for doing the Western blot. I think this is the most convincing thing here, is to do multiple proteins from one sample, just do different injections. You can also run multiple samples off of one chip and then run them in parallel, which I think is a bit difficult to do right now, but maybe could be done eventually. Now, the latest thing that we've done with this is thought about trying to get away from that dragging of the membrane, the capillary or the chip across the membrane. And one of the reasons for that is it's very doable, but you do have to be a little careful about lowering the chip down to the membrane, getting about the right depth. If you go too hard, you create problems where the proteins don't adhere very well. And then if you're too far away, they spread out a lot and you get a lot of spreading. So there's sort of a narrow window there. So we got into this idea of using inkjetting to deposit the membranes. And so what we put at the end of the capillary, this is going to be with capillary electrophoresis now, not chips, but put this in here, and then just let an inkjet printer inkjet like you have on an inkjet printer will eject little droplets very rapidly, 50 picoliter droplets out. And so those can be almost any distance from the membrane. It's very, very reliable in terms of distance. This works quite well. Here's an example of looking at the, let's see, this is a Western blot here, and two different proteins in the same sample being looked at, and then here's the library down here with that type of setup. And one of the things that Peter did after he had this is start to look at smaller samples. So in this case, he was taking 3 islets, so about 9,000 cells, processing them, just nothing fancy, just small vials, and loading that onto the chip or the capillary for separation. And here's, he was able to get numerous assays. These are all done from one 3-islet sample from this. He wasn't able to detect all the proteins that we hope, but I think about 9 of those were being detected in that particular case. So you can do multiplexing from very small samples that way. Okay, so everything I talked about is CE because this is CE Pharm, okay? But it's all CE and no Pharm. So I thought I would tell you about some work that we're doing lately with HPLC, but actually does have a Pharm application in it, okay? So I just change gears a little bit. So one of the things about CE that I always thought was really cool is those second, sub-second type separations, okay? And it's like, you can't do that by LC. And actually, it's not true. You can do sub-second separations by LC. They're not as highly efficient as you can get by CE, but you can do it. And so Dan Armstrong, showed that you can do this. He was taking guard columns and, 0.5 centimeters long, and, they're almost as big. The diameter is almost the same as the length of these little columns. And you can get, here's a 1-second separation here of these guys. And then here's 4 compounds resolved in 1 second by doing this. And there's no real trick to that, just short columns, high flow rates. These are mils per minute flow rates, so these very short columns. And you got to minimize your dead volume or you get nothing. But you can do this. Now, the problem is, what's the point? I can put this on an autosampler, and my autosampler can take its sweet time and take 45 seconds to a minute to do the injection, and then I get a 1-second injection, right? So it's not high throughput, okay? And Jim Grinius, who's my collaborator on this project, sort of demonstrated this. They sort of messed around with the autosampler and got it down to, I think, about 45 seconds. And, some are a little faster than this. He was doing about a 15-second separation here. So that's kind of a nice throughput there, decent peak separation, but it's still not as fast as the 1 second, right? So we started thinking about how we could actually make this a high throughput LC. And this gets back to the idea of using droplet microfluidics. So our concept is that we're going to get rid of the autosampler, and we're going to have our samples configured as droplets, again, separated by this immiscible fluid like fluorinated liquid. And the idea would be you pump that droplet train continuously into the injection valve. When you fill the sample loop, you can inject it. Now, by the time this you reactuate the valve, the second droplet is already there. So it's really kind of minimal moving parts, and you're not having to wait for the autosampler to move around to do and rinse a lot of things out. Does it work? Well, here is an example of doing 96 separations in 96 seconds, 3-component mixtures. You can see the resolution down there. So granted, these were picked to, we had to find the conditions to get all 3 of those things separated, but you can inject pretty fast. This is going at 3 to 5 mils per minute at a little elevated temperature with this approach. This is 2 seconds on the left. So we've done a lot more with that, but I just want to sort of jump ahead to talk a little bit about, maybe starting to think about using fast LC in different ways. And one of the things that is easy criticism of that is like, oh, your peak capacity is terrible, right? Peak capacity is 3, 4, 5, something like that, maybe 10. So if we slow things down a little bit, we can get a pretty decent peak capacity if we run a 15-second gradient. So you can see that's what the chromatogram would look like. This is from a peptide digest. And then if you want to get a higher peak capacity, what if you couple it to another very fast separation like ion mobility? So this is what we did here. So we are doing now 15-second separation interface to ion mobility separation with mass spectrometry. The peak capacity just on the 2 separations is about 375 with this. That's pretty good for a 15-second separation, I think. So one area that we thought we would look at is to use this to characterize antibodies by digesting them and looking at the peptide digest of your antibody. Devin in my group was collaborating with Brandon Rutlow's lab, looked at this and showed that he was getting something like 96% sequence coverage from the antibody in that 15-second separation. If he slows it down, you get up to 99% for this antibody. And he further went ahead and showed that you could detect effects of stress on the antibodies. So here's an example where we had the control run, looking at the M/Z range for this particular peptide. This is what the LC looked like for that. That's what the ion mobility separation looked like for that peak. All fine, 1 peptide, 1 peak in the LC, 1 peak in the ion mobility. After the high pH stress, though, the LC looked about the same. Remember, it's a 15-second gradient. You don't get a lot of separating power. But the ion mobility shows you actually 3 peaks now, which he was able to show that one of them was a +1 Dalton deamidated, and the other one was some sort of isomer from this peak. So you can detect these stressors on these antibodies and their effects with that very quick separation. So I think the point of this is that the realm of very fast separations is not just GC and not just CE anymore. There's potential to do it with LC as well. So with that, I want to close. I want to thank you all for your attention. I also need to thank the group here. These are the current students. Peter's the one that did the Western blotting that I showed you, and I think Devin was not here that day, so he's not in that picture. And then I want to also thank some previous group members. So this is a group when a lot of this Affinity CE was being done. And you might recognize this person right here in the Cheeto. I wasn't going to point it out until your introduction. So I will now point out that in her Cheetos t-shirt. But Kristen was super fun to have in the lab and was, as she said, very passionate about our science, which is something that I absolutely loved. So with that, I want to close, and again, thank you for your attention.
All right, thank you. All right, so now it's time for questions. So you can either come to the mic in the middle. If you want to raise your hand, I need to get my steps in so I can come give you this mic. Okay. I'm cold and I need to.
Thank you. It's a really nice talk. I just have one question about your binding studies using the CE. I'm just curious, during the binding partners, if one of the protein is modified, have you got a chance to see how sensitive is the method to pick up the differences? Let's say if one of the binding partner has a modification and it has an impact on binding. That's my first question. And the second question is, were you able to compare the binding constants with any other methods?
So I can answer those questions a couple of ways, I think. So let me. Oh, I see. Okay. So here's an example that's an exact example of what you're talking about. So here we are looking at some modifications to proteins. You know, this is the wild type HSP70, and then these are single amino acid substitutions that are being, or no, excuse me, 2 amino acid substitutions that are being done on HSP70 in 2 different ways, and that affects the binding curves that you get, okay? So that's an example where I can say, yes, you can detect changes in binding affinity due to modifications of a protein. Now, a better answer would be to say, how small of a change in KD could you pick up? And that would require some sort of mathematical analysis of what the difference, you know, how much error you have in KD compared to there. And I don't have a number for you there, but you know, these are pretty small shifts that you can pick up here, as you can see. And then you had 2 parts to the question. And what was the? Yeah, I've forgotten it. Oh yeah.
With any other methods like BIACOR?
Right. So we did do a bunch of comparisons, and what I should tell you, I went over this a little quickly. So we had some of these we compared to this fluorescent cytometry assay that Jason was proficient with. Some of them were by some sort of calorimetry thing that they were using as well. Some of them are from the literature and done by BIACOR or surface plasmon resonance. So, you know, we didn't do a head-to-head comparison of numerous protein-protein interactions this way to BIACOR specifically, but it's represented in this graph because some of the, you know, this is a very generic term, non-cross-linking method. So some of them were BIACOR, some of them were calorimetry methods, some of them were the flow cytometry method. But they were, in general, we were getting good agreement across there. And I would expect to get something similar to BIACOR. Yeah.
I have a question about the affinity CE, right? Just now you mentioned about there are 3 challenges about the affinity CE. You worry about the on-column dissociation between the protein and the binding compounds. But the third challenge is about the interaction of the protein with the capillary surface. This can somehow mess up the, you know, the binding interaction between your drug and protein. Do you have a universal coating to address this challenge?
Yeah, so the question, that's sort of like I think the thing that really holds this method back is if one of the binding partners interacts with the fused silica or glass surface that you're using, that's going to affect what you see, and that's totally true. And I think that we would spend a lot of time trying different buffers, you know, these goods buffers, and pushing the pH as high as we could without affecting the binding, things like that when we were doing just the free binding. And we often could find good conditions, but it was, I would say, a slow process. The cross-linking method worked much, much easier. We could bind in whatever solution, cross-link them, and then we could just, you know, do whatever we wanted to those proteins, and things were fine. But of course, that's not good if you're looking at a small molecule binding to the protein. So I think there you're a bit stuck unless can do some kind of competition. If you could have something that was fluorescently labeled that you were competing off, or the protein, yeah, I think that's what you want if it's a small molecule. Sort of like what we're doing with immunoassays, then you can probably get at it that way. But I would say that's probably not the best use for CE just because of that challenge. Yeah, definitely a problem. Yeah, sure.
And then when you have a question, just say your name and your company per CAS tradition. I forgot to mention that.
Okay. Yes, my name is Göran Hübner from Birmingham, Southern Germany. Thank you very much for the great talk. I've got a question, even though it's a CE community right now. Your last topic regarding, let's say, high-speed separation and of peptide. So I'm trying to set this up with a chip-based CE system, and I'm also, trying to include ion mobility into this to exactly do the same stuff. To even if I get no separation of isospecific peptides from wild-type peptides in CE, to try add a second dimension there. What type of instruments did you use, and did you tweak the ion mobility settings in order to get it separated, or it was just as it is, without any optimization there?
Yeah. Good question. So it was a Agilent LC, and the ion mobility was a Waters cyclic ion mobility instrument. So what you can do there is, one of the main things you can adjust is the number of cycles that you let it go through to get separation. So I think because you don't know what you're going to have, Devin just, I think, set it up for something that was reasonable to be sure that he got enough peaks across each chromatographic peak. What I know is that it didn't take him long to optimize it. I'm sorry, I don't have all the specifics about that. He's still working on the write-up for me, but what I can tell you is it was much harder to get the software from Waters and do the data analysis than it was to get the separation. Yeah.
Hi, Bob.
Yeah.
Also, Richard. Thank you very much for a very good presentation. Give you an overview application about this Affinity CE. I don't really have technical question, but I just have a question regarding this. What do you think about this technology in the future, move forward, especially to you and also the vendor? Because you can clearly see, the technology is much better than the current flow cytometry, or the ELISA, or anything on the bioassay that you've been looking up and down, and in the separation, you have a second dimension, you can demonstrate that, we see that the opportunity, and you increase the separation power really fast with a high throughput. So what do you think we should do in the community as a separation to sell this technology, and maybe bring up some vendor to bring this up, so we can compete with this more traditional ELISA or flow cytometry? I really think the separation technique is the best technique to do a binding assay.
Yeah. If I knew that, I would probably be coming to you as a vendor seminar, right? So I think it's a great point. One of the things that I know about analytical instrumentation, though, is that if there's something established that works pretty good, it is very hard to displace that with something, even if it's better, if it's very different. It's just really, really hard. And I think that you can see, given what we talked about today, that the protein simple is a good example where that product, if you can afford it, and the materials cost, is a lot better than a regular Western blot, right? So it was a big enough difference that people were willing to make the leap, again, if you can afford all the materials costs and everything. So I've been a little frustrated by this myself, is that the CEs were put together to do a certain kind of experiment, and even the chip CE put together to do a certain kind of experiment, and they didn't take, I'm sorry if there's vendors here, they didn't take advantage of all the advantages of CE. Like, they're not built to do really high throughput CE, for example. So But I think there have been some advances since some of these instruments were developed, like the droplet introduction techniques. I mean, I showed you one, but other people have done different things. And so I think you need a good vendor, because I think the problem is that the people in the industry don't have time to fiddle around with these instruments the way that my grad students do. I know that. But so you need a good vendor for some of this. Now, the protein cross-linking thing, I think anybody can do, right? That's just not hard. And that is something that you could maybe start to do. But it's going to be hard to displace these established techniques that are good enough for most people, right? So that was kind of a rambling answer because I don't know the answer. Yeah.
Just name your instrument the Bob.
Maybe name it Kristen, because it always works.
It always works, yes. I will take great pleasure if you name something the Kristen. I have a question about the body on a chip.
Yep.
So knowing what I know about you and what was done in the past and now, I remember putting the adipocytes on with the islets and seeing everything. Do you see, then I'm remembering what John did with his, dinner plate,
Yep.
high throughput. Do you think your lab has the capabilities to do a true body on a chip where you have 3 or 4 different systems on there and perfusing and then going into that poor little islet being like, oh dear God, what am I seeing right now? Are there any limitations? Do you think you will get to that body on a chip?
Yep. So I think let's see. Other people have done it, so I think we can do it. All right. But I do think that the trick is integrating the sampling side to do the analysis. But one of the things I had trouble with is sort of thinking, what's the question I want to answer by putting more organs on the chip? And I will say that when it comes to endocrine systems, I think fat plus endocrine plus liver, because the islets act directly on the liver to affect glucose production, and then maybe muscle, because muscle also releases hormones that affect these things. I think that would be a good combination. You can imagine varying amounts of the adipocyte to muscle to vary like sort of percent body fat, for example, would be a pretty interesting experiment to do. And yes, I think we can do it. I just need the will. Yeah, that's right. Yeah. You want to come back and do that?
I have a couple.
You probably have a job, I know, but
No, I don't do chips. I don't touch chips with a 10-foot pole. All right, anything else? Any other more questions? Oh, yep, one in the back. Is this even on? This is,
it's working.
It is? Okay.
Yeah.
I think with the industry really pushing like automation and AI, I know you mentioned that for one of the projects you were looking at a couple thousand different electropharograms. How do you think automation and AI would have helped with some of your experimentation?
Yeah, that's a good thing to be thinking about. I think one thing that AI might be able to help you with is finding conditions, CE conditions for looking at interaction partners, right? Just like we were talking about before. I didn't think about that, but potentially if you've got a database of work that you've done and tried different things, and you can also pull in from literature, AI can maybe help you make sense of that and try to find conditions that would allow you to get good CE conditions for, say, rapid separation of a particular complex. For example, you put a few experiments in or a few things you know about the system and see what you could get out. And I certainly think, and I'm sure people are doing this already, is if you've done a screen like I showed you and you get, the properties of all those compounds and the ones that are hits and the ones that aren't, that you could sort of use that to help design better inhibitors. I mean, that's something I'm sure people are already thinking about and doing. So those things really help you build up the databases that you have. Now, it turns out that the data analysis that we did, which I think was maybe a part of your question, was actually not too hard because I had a student in my lab that developed this program that lets you look at all the electrophorograms at once, and it could basically very quickly mark them all for complex and free peak determination really easily because, again, the reproducibility and migration times we have was really good, so it's just knew where to go look, and it gets through them all very fast. So that part has always been kind of easy for us. But I think making sense of the data when you're done, that's where AI can be really very powerful. Yeah.
I was going to be the auctioneer, but Rick, you stopped me.
Well, someone has to stop. Rick, I just work for AstraZeneca. Since actually my boss, unfortunately.
Okay.
I'm kidding. I love working with her. So for the LC assay you were talking about, obviously it's fantastic that we can get down to that 15-second separation time. But I know, at least for us, a lot of the high throughput portion of it is how much analyst hands-on time there is for sample preparation. So when you're loading that microfluidic capillary for sample, is that significantly more analyst hands-on time than prepping the autosampler, or is it easier or comparable in some way?
Yep. Great point. And I would say that the way our first paper we published on it, the way that droplet thing worked was not what you would even want to think about because it was, getting the droplets ready was not a particularly fast step. But we've got this now where it's completely automated. So you can have a regular well plate, sit it down in front of the instrument, and it'll just sip up the droplets and inject them automatically. So it's pretty easy.
Okay.
And then the other thing is, there are tools for doing the sample prep for the digestion as well. So we were using a Waters kit that's pretty slick for doing pretty quick digestions and doing that in parallel. So that part was also reasonably fast. So that's definitely coming along and getting more and more automated. Yeah, but I know what you mean. You got to have the whole thing automated to make it easy. But that is something that we actually are doing, and we're basically there now.
Okay,
so
great. Thank you.
Yep.
All right, anything else? Going once, twice. All right, let's thank Bob for a great keynote.